Hi everyone! Thank you for watching the Earth Life Science course orientation video for the April 2027 admission. The ELSI course orientation consists of two parts. First, this YouTube session that you're currently watching, and second, a live orientation webinar which will be held twice on July 24th, Japan time. Please be sure to check the time for your own time zone. The orientation webinar requires registration. If you haven't registered yet, please visit the QR code on the right to complete the registration process. In this YouTube session, we will begin with an overview of the ELSI course, followed by presentations of research activities by ELSI PIs and assistant PIs, and information about the application and entrance examination process. So let's start from the overview of the ELSI course by Professor Sean McGlynn.
Hey everybody, my name's Sean and I'm one of the ELSI faculty, and I'd like to describe for you today about our institute and the place of the institute within our university, and then go through some of the basics of the graduate course that we've created in the last couple of years at the institute. So the institute that we're at here is called the Earth Life Science Institute, or as we usually say, ELSI. And we're housed at the university called the Institute of Science Tokyo, which was created recently through a merger of the previous Tokyo Institute of Technology and the Tokyo Medical and Dental University. So we have a new university that's more comprehensive than previous, and we call it Science Tokyo for short. So we're housed at Science Tokyo, and we are part of one of the world premier international research center initiatives, or WPI for short. And these are rather unique programs that are created through the Japanese Ministry of Sports and Science and Education, MEXT, or Monbosho, as we say. In these research centers, you can see a map of them shown on the screen. And these are institutes that are created within universities, focusing on a specific topic. And so you can see the variety of the different institutes here shown on the screen. ELSI, the Earth Life Science Institute, the focus is the origins of planets and the origins of life. And we kind of have one major question at our institute, and that is, how do planets make life? And to address that question, we are researching across multiple themes. Of course, to understand how planets make life, we have to understand how planets form and why they form the way they do, and then what are the molecules and atoms and abundances on the planet, and so on and so forth. So it's a very interdisciplinary research avenue that we're addressing here at the Institute. So I mentioned it's interdisciplinary, and it is. So we have people all the way from planetary scientists, numerical modelers, geophysicists, chemists, microbiologists, biochemists, and so on, all trying to work toward this topic of how do planets make life. And so this is an institute. It's not a chemistry institute. It's not a geology institute. It's really its own thing, where we're all in one building, all together, and the kind of pillars of the WPI framework are shown on this screen, where we try to do top level science, and we try to meet together with people that are from traditionally different and separated fields and work together. We try to do that in an international environment, and we also try to do that in a way that's perhaps a little bit unusual or different from the typical Japanese research environment. So there's an opportunity to reform and perhaps work across or through or transform hierarchies into a more collaborative and open research activity. So within our institute, we've started a graduate course. We call it the ELSI course. And this is an integrated master's PhD program, which operates between three different schools: the School of Science, the School of Material Science and Chemistry, and the School of Life Science and Technology. And so students can enter into any of these three different schools and then participate within the ELSI course, which is this integrated master's plus PhD program. And the goal here we have is to have this broad educational opportunity focused on astrobiology, the origin of life, the question that we have, how do planets make life? This is a 5-year program in English. We have financial support, and I'm going to get into an overview of how this works in the next slides. So here we are on a beautiful spring day around the 1st of April at the Institute. The sakura are blooming, and we can talk about the details of the course curricula that we have here. So let's go ahead and kind of go inside and check that out. So the overview of the course here is really thinking about this main question: how do planets make life? And we want, we're recruiting, we're interested in having students that are interested in these fundamental questions in the natural sciences about how do planets form and how does life form. We also think that addressing these fundamental questions has a strong relationship to current contemporary issues, environmental climate issues and resource distributions and so on, sustainability, not only on Earth, but life and sustainability on other planets. Shall we go there? So, as I've mentioned before, this is a 5-year program. Students all do a master's. There's a formal master's component. Students write a thesis, have a master's committee, and then graduate from the master's, but then immediately and smoothly enter into the PhD component, which is a 3-year term. And so by doing this 5-year program in an integrated way, we're seeking and we're hoping that students will be able to ask big questions and have enough time to ask those big questions. And part of being able to ask these big questions and have the kind of freedom to do that is that we supply financial support. It's about 170,000 yen per month. And this is, it's enough to live here in Tokyo. Tokyo is a big city. Sounds expensive maybe, but compared to other cities, if you think about New York or London, it's actually very reasonable to live here in Tokyo. So thinking a little bit more about the finances here, so students have a scholarship, this 170,000 about yen per month, and then it's worthwhile to note and to think about a budget. Shall you come here? There is an enrollment fee that the students are required to pay, and there is a tuition fee as well. And so you want to put these numbers into a sheet and think about your financial situation. And it's worthwhile saying that the tuition fee for the subsequent semesters, it's possible to have them waived. And please talk to a potential faculty advisor about the waiving and how this works. And also, like I mentioned, it's a good idea to make a budget, and you can do that with a potential faculty member as well. So what's going on scientifically at the Institute? So we're researching across different themes and approaching this big question about how do planets make life. And to do that, as I mentioned, we're not a chemistry institute, we're not a geology institute. We're really working across different themes, and we have expertise in multiple areas. Not all of them are listed here. Here's just a potential list or a small list of the expertise that are represented at the Institute here in green on the right. But you can think about, researchers at the Institute all in one building are thinking about solar system processes all the way through biomolecules and polymer-driven replication and evolution with the goal of really creating new research through the fusion of disciplines that are usually separated from one another. And to do this, part of the way that we do this is we have open mixing space. And so we have, for example, the ELSI Agora. Here it is on the 2nd floor of the building. We have this open space where students, faculty, researchers, postdocs, so on and so forth, can all mingle and talk and run into each other and ask questions. And we're really encouraging the asking of questions. And to do this, this is part of why we've created this graduate course program, is because being a student helps us ask questions, and that's part of our goal here, is to have this open questioning environment so we can have this fusion in the research. So it's about 50-50 Japanese and international. The official language is English. It's kind of, it's so international. Sometimes I think of it like a study abroad that's happening here right in Japan, or ryugaku mitai in Japanese. And but the English language is the official language. So your lectures are in English, the thesis and the presentations are in English. And so we have this international environment right here in Japan. In addition to the academic environments, we recognize the importance of having students be involved with, potential industry opportunities and industrial research opportunities, collaborations. And so we've got an opportunity for students to do internships, to be involved with companies and including energy and space industries. And we want to create an environment, opportunities for students to be able to work on, not only in academia, but potentially move into a more applied type of a work career later on, should you choose. So what's going on in the course? We've got a master's course curriculum where we have foundational courses in chemistry and biology, planetary sciences. These are, again, lectures in English. And these foundational courses are really addressed at students who are coming to the Institute from multiple backgrounds and multiple disciplines to give you the basic vocabulary and tools to be able to communicate with one another later on. And so you might have a background in geology, you don't know anything about protein biochemistry. By the end of our biology course, you will know the basic vocabularies and workings of enzymes and proteins, for example, and vice versa. You might come from biology, but you don't have a background in planetary science. You don't know that vocabulary. By the end of this course, you'll have that vocabulary and you'll be able to start asking questions about that field. Again, with the goal of being able to achieve this fusion research. So here are the courses we have: chemistry, biology, planetary science, taught really kind of at an undergraduate level, kind of an introduction to diverse students, because we're accepting students from multiple disciplines, to give students the vocabulary you need to be able to talk to each other across these fields. In addition to these basic courses, we've got our industry collaboration course, and we've also got our global communication class, where students are equipped with skills to help you become more effective communicators, uh, with a special emphasis on public communication to the public. In the doctor course, the focus is more on research, less on courses, and you are really focusing on the development of your doctor course thesis research. And again, you have an opportunity to pursue internships as well.
Next, each PI and API will provide a short video explaining their research activities and interests. Only the PIs can be your official supervisor, but we also encourage you to work with APIs.
I'm Yasu Sekine, PI of ELSI. Is there life beyond the Earth in the solar system? This is the fundamental questions we have been tackling in the last several years. So, according to the recent advances in the spacecraft explorations, we now know that there are multiple habitable planet or habitable satellite in our solar system in terms of the presence of liquid water, organic materials, and also chemical energy. So according to the recent Mars explorations, we have known that there are barren network clay minerals and sediment on the surface of Mars. And currently, the Mars rovers have been rovering on the surface of ancient lake sediment and collect the sample rock to analyze its chemical and mineralogical composition to reveal the habitability of this planet. And in the outer solar system, there are multiple geologically active icy ocean worlds where the subsurface ocean exists beneath the icy icy surface. So spacecraft now did the in situ analysis for the fluid materials erupting from the subsurface ocean and analyzed its chemical composition to reveal whether the subsurface ocean environment is really habitable for life. So in my laboratory, the main research methodology is laboratory experiments in which we try to reproduce the physical and the chemical conditions like temperature, pressures, and radiations on the planet and satellites, and to understand the chemical reactions and the geophysical processes possibly occurs on these planetary bodies. And we try to compare the results with the space exploration. And in the collaboration with field geologists and also the synthetic biologists, we try to do the new researches like to predict the potential biomarkers in the planetary body. And also we try to do the field work to the terrestrial analog field of Mars and IC planetary bodies. So I have been involved in several space missions, international space missions, which include the International Mars Ice Mapper and JUICE, Jupiter Icy Moon Explorers by NASA, by ESA, and also Dragonfly mission to Titan, led by NASA. So if you're interested in these international space explorations, then I also welcome you. So currently, we have almost 10 students plus one postdocs, and we welcome you to join our group. Thank you.
Hi, konnichiwa. I'm Hidenori Genda and one of the PIs at ELSI. The solar system has many different types of celestial bodies, such as planets like Earth, moons, asteroids, and more. Each one has unique characteristics. For example, the size of planets are different, and even Venus and Earth, which are similar in size, have completely different surface environments. Our research aims to uncover the processes that have shaped these diverse features. By conducting these comparative studies, we hope to understand the universality and uniqueness of Earth's characteristics, such as why life was able to emerge here. Sometimes, or rather often, it's challenging to conduct direct experiments in the laboratory. For example, this figure shows a giant impact that is believed to have formed Earth about 4.5 billion years ago. What happens when planetary sized bodies collide violently? What kind of surface environment emerges after such a collision? Our group performs computer simulations considering the physical and chemical laws. Here is another example. A simulation where we gradually removed Earth's oceans. This helps us explore how the amount of water influences the stability of a planetary climate and whether Earth was truly the ideal environment for the emergence and evolution of life. Almost everything you can imagine can be recreated in computers. However, it's essential to validate the outcomes obtained from those computer simulations. Therefore, our group conducts our research with ongoing and future planetary exploration missions in our mind, collaborating with observational teams and experimental groups to validate our findings and hypotheses. Currently, we are a group of about 10 students working on various topics related to planetary science. We'd love to hear your ideas and work together on exciting research. If you are interested, please visit our group's website and get in touch with me directly. Thank you.
So my research group is interested in how planets evolve and how they're shaped by dynamical processes that happen through the entire planet. And this includes processes from the center, including the cores of planets, how magnetic fields are generated, to mantle convection in the rocky parts of the planets, plate tectonics, volcanism, as well as the evolution of the surface environment. So what we're trying to do is look at planets and have a deeper view of the interior to understand how these planets are working, how they're exchanging matter throughout the entire system. And it's a very much a system science approach that involves the entire planet from the center to the surface environment. To do this, we use models. And in my group, we use both forward and inverse models. So forward models are simulations. So we will simulate mantle convection and plate tectonics and things like that using computers, similar to what Genda-san's group does. And but we'll also do inverse models where we'll collect data such as seismological data, gravity, or geomagnetic data, and we'll also use models to invert for the properties of the interior of the planet. And our goal is to understand how the planets behave as a complex system from their formation to through history, and how this couples then with the biosphere, the emergence of the biosphere, which is a part of the geological system in our view. So that's all for my side.
Hello, everyone. This is Takanori Kodama, one of ELSI APIs. My laboratory just started since last October. My biggest question is, what is the Earth? This question is a general question related with not only a science, but also various science subjects, which include astronomy and astrobiology. Our laboratory tries to understand this question based on planetary climate system. The climate system consists of several subsystems, which include atmosphere, ocean, continent, and ice sheet. For example, clouds play a significant role in the climate, and atmospheric and ocean circulation transport water and energy. Distribution of continent controls ocean current, planetary albedo, and evaporation rate of water vapor. Ice sheet has higher planetary albedo, as you know, and important effect as storage of liquid water. So, planetary surface environment is a result from climate system. My research key questions are: what kind of climate system does the planet have? And what is a habitable condition? Additionally, what did and will happen as a result of climate system change? In the climate system, there are many physical processes, which include radiation process, atmospheric and ocean circulation, and precipitation, and so on. Actually, they have various different timescales. We model these systems numerically and develop three-dimensional global climate model. Depending on science topic, we use different models. Recently, we started to develop the couple atmosphere ocean global climate model and global cloud resolving model to apply to different environments with the Earth. Thanks to computer resources, we gradually access to supercomputer like Fugaku and Earth Simulator 4 at Jamstack to run our simulation. Our research topic are climate and habitability of extraterrestrial planet like TRAPPIST-1 system and and solar system planet and moons. I mean our Mars and our Venus climate and subsurface ocean on icy moons. And also we started paleoclimate event like snowball state and also mass extinction, which destroyed the dinosaur world, for example. There are many open questions in my laboratories. If you are interested in our research and our research topic, please contact me. Thank you for listening.
Hi everyone, and thank you for watching this introduction to the Nakamura Lab at ELSI. So one of the big questions we are exploring is, how did geochemistry become biology? So this is truly fascinating and a fundamental question directly related to the origin of life. More specifically, our research focuses on understanding the transition from geochemistry to the early stage of biochemistry. To tackle these challenges, we aim to bridge the gap between geology and biology by utilizing and maximizing our strong expertise in material science and catalytic chemistries. Among the many geological environments so far proposed as a plausible setting for emergence of life, submarine hydrothermal vents are our primary focus, as shown here. Why hydrothermal vent? Because the chemical reaction and energy conversion processes in this system are quite similar to those found in modern cell. And in our lab, we are one of the few research groups in the world that collect real natural hydrothermal vent sample from deep ocean and analyze their chemical and physical property in quite detail. And based on our finding, we have shown that hydrothermal vents are not just a simple chemical reactors, they are in fact electrochemical electrosynthetic system where electrical current generated across a mineral membrane can drive carbon metabolism. Even more exciting thing is recently we discovered that ion gradient in this system can also be converted into the electrical current, just like living cells are doing now. Currently, we are pushing this model forward to experimentally demonstrate the emergence of protometabolism and primitive cells. If you would like to learn more about our research, please feel free to contact me. We are always excited to connect with people who are curious about the origin of life and the mystery of Earth's natural system. Thank you.
Hi, my name's Harrison Smith. I'm a specially appointed associate professor here at ELSI. My research themes revolve around the origin, evolution, organization, and detectability of life. So the key questions that kind of come from that have to do with how to distinguish living and non-living systems. For example, you can represent living non-living systems, chemistry and biochemistry using networks, and ask questions about how those networks look topologically and scale over time. How do you look for life outside of Earth? So these have to do with questions of what are the best techniques to look for life outside of Earth in a reliable fashion. And relates to the previous question because you can ask questions about how hard is it to distinguish planets which have biospheres and planets which don't have biospheres. And then what was the evolutionary trajectory of the biosphere and biochemistry overall, and what constraints guided that evolution? So I'm investigating this by looking at kind of the history of reactions and metabolism and how those change over time. A little bit about me as an aside. I have a BS in physics and astrophysics from the University of Michigan, did some cosmology research, brief stint as an engineer at NASA, Did my PhD in astrobiology with Sarah Walker at ASU, and then I joined ELSI as a research scientist, and I started as a specially appointed associate professor this year. I'm also affiliated scientist at Blue Marble. So the kind of methodologies and techniques that I use are all computational, having to do with bioinformatics, for example, pulling information from these biochemical databases online, layering on things like thermodynamics, applying complex system science approaches, so network analyses, information theory, Bayesian probability theory, agent-based modeling, etc., representing things as networks or doing, again, things like agent-based modeling. And these can be static or dynamic approaches, so we can talk about doing kind of topological and scaling techniques, but we can also talk about running simulations and looking at time series. So with that, my brief introduction is over, and I ask what interests you. So if you've read any papers, find them interesting, had thought experiments or research ideas that were provoked by those papers, please feel free to let me know if you're interested in collaborating. The kind of skills that I use and that you would learn and develop here are related to programming, data analysis, and statistics. And again, thanks a lot, and reach out by email if you have any questions about working together with the program. Hey everybody, my name is Sean, and I'd like to take a few minutes today and describe about the research activities that are happening in my lab here at ELSI at Science Tokyo. So in the lab, members are working on lots of different topics, ranging from chemistry through enzymology, through microbiology, and all the way into field work, where we're focusing on hot spring sites, which might be similar to some conditions in their early Earth history. And across all of these different research avenues that group members are pursuing, the central question that I have in my mind that unifies all of them is, how is chemical organization accomplished by energy flow? And I ask this question because, in my mind, biology is an example of material organization that's accomplished by energy flow. And I think that by studying these so-called different disciplines of chemistry, enzymology, microbiology, and even fieldwork, we might gain an understanding of kind of general rules and principles that govern organizational processes as they occur through the transmission of energy. And so we're not going to have enough time to go into great detail today. This is more just a way to say hello. And part of that is introducing you briefly to the group members. Here's a picture of the group as it was last April in 2025, and just after the sakuras are blossoming. And what I've done here is just listed out different group members' topics that you can see and view the diversity of research activities that are happening in the lab, ranging from kinetic isotope fractionation of enzymes through polymer evolution and energetics of archaea, group transfer potential chemistry, and so on and so forth. And so you can pause the video and take a look at some of these topics. And the thing to think about here is that we are not a lab that does one thing or centers around one thing, but we have this big question in our mind. We want to understand how material is organized by energy flow. And so to do that, we really need to study different types of energy and how energy is harnessed physically and chemically. And that's why we're working across all of these different scales, all the way from small molecules and elements, the subject of traditional chemistry, and on upwards. And so for your interest, to talk more, to learn more about the research activities, please send me an email, and we can have a discussion. Tell me what you're interested in, and we can open up some of these topics and explore together. Thanks a lot for your interest in the ELSI program, and thanks also for your interest in the lab.
Hi, I'm Liam Longo, a specially appointed associate professor here at ELSI. In my group, we are interested in the evolution of biological complexity, and we take inspiration from a quote by Darwin. From so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved. To uncover these simple origins and their connection to life today, we use a combination of experimental and computational approaches. Here are some selected publications from the last couple years. Because time is short, I will introduce you to 2 key questions that my lab is working on to give you a sense of the type of research that we do. In the first project, my group tries to understand how ancient geochemistry gave way to modern biochemistry. What I am showing you here is a computational approach that explores the evolution of biochemistry. In this trajectory, the reaction network is being built out step by step as new compounds are being discovered. Approaches like this can help us understand where forgotten or missing biochemical reactions may be hiding. I've been captivated by protein structure since I was a graduate student. In my lab, we try to understand where complex molecular structures, like the one shown here, came from. To our surprise, we found that structures like these may have come from ancient peptides that form tiny droplets when mixed with RNA. This work reveals unexpected transitions in protein evolution. Finally, I'd like to tell you what skills you can learn in the Longo lab. Content-wise, we integrate biophysics, biochemistry, and bioinformatics. In addition, we put a very strong emphasis on effective writing and communication. There are several open projects in my lab, so if questions like these sound interesting to you, please stop by my breakout room and we can discuss more. Bye everyone!
Hello everyone, my name is Kosuke Fujishima, and today I will give a brief introduction of the Fujishima lab. Our lab is currently focusing on using synthetic biology to tackle the fundamental questions in the field of astrobiology, such as origin of life. Origin of life is yet to be solved fundamental question in the field of science. In order to tackle the origin of life, we want to understand where the functional biopolymers came from. As you may already know, Earth life uses DNA, RNA, and protein as the core polymer in the biological system. DNA and RNA serve as a genetic information carrier, whereas RNA and mainly protein serve as a catalyst. The question is, where and how did these polymers came about on Earth or in other planetary bodies, such as icy moons or Mars? To tackle this question, we are currently taking 3 different approaches. One is prebiotic chemistry, to understand what sort of organic compounds or bio-related compounds can emerge in a certain environmental settings. Secondly, we use computational approach to understand the interaction and behavior of molecules such as amino acid or nucleotides in relation to the membrane. Thirdly, we use synthetic biology in order to create a protein or RNA molecule that could have existed in the past. So our lab basically spans from origin of life all the way to synthetic life, where we can make a lifelike system. Just to give a brief introduction to the synthetic biology research, our lab core technology is mRNA display method. This method allows us to screen for a large number of protein, up to 10 to the 12, and screen these proteins under different functional screening inside the test tube. These proteins could be either consist of a limited set of amino acids or a completely de novo sequence, which life on Earth has never been explored or used. So we create a library of these proteins to screen for key functions, such as RNA binding. The other approach is the prebiotic chemistry. For instance, our lab is focusing on a unique environment in the deep sea, so-called water and supercritical CO2 two-phase system. This environment allows water to evaporate and concentrate in certain molecule. And also, this environment creates a very acidic condition where phosphorus could be obtained. Therefore, we were able to showcase nucleotide formation under this environment using a high-pressure reactor. If you join our lab, you will also learn how to analyze key biological compounds using ELSI's analytical instruments, such as LC-MS, shown in this picture. We also have other analytical techniques and tools available, so we're happy to explain and provide guidance to how to use these techniques and instruments. Last but not least, our lab is very international, and we're very open to new members. We also collaborate with various researchers all around the world, including researchers from Czech Republic, USA, UK, and Singapore. So if you're interested, please contact me through fuji@elsi.jp email, and I look forward to talk to you. Thank you.
Hi, this is Tomo Matsuda, one of the faculty at ELSI, Institute of Science Tokyo. These are my group members, 2025. Our research interest is to build life or living system from scratch. And in order to achieve this, we work together with many collaborators inside and outside ELSI, and also in collaboration with many international collaborators. So in a few minutes, I'd like to explain the background of our research. As I said in the beginning, we're aiming to synthesize life, but if you think about the origins of life, it's very likely that the life has started from very simple compounds like methane, CO2, and hydrogens, and so on. And through the course of chemical evolution, it has become much more and more complex, resulting in the synthesis of amino acids, nucleic acids, or fatty acids that are the components of the current living system. And these molecules, in some way, were assembled into a system so-called molecular system, so-called a protocell. And the evolution is very likely to bring this kind of protocell into current cells which we see right now. So the important steps in the origins of life is chemical evolution and biological evolution. And in our research group, we mostly focus on the upper part of this schematic, where we assume that the molecules or biological molecules are already present, and ask how these molecules can be assembled into a molecular system. So more precisely, we work on molecular system assembly to reveal the recipe of a living system. We also work on molecular system assembly in geologically possible, plausible settings. And we also work on directed evolution of proteins and RNAs that at the end will be used to construct our artificial cells. So our research group have 4 research teams, so to say, one of which is the Origins of Life research group. And here we're using various environmental oscillations to assemble molecular systems, or we can call it as an artificial cells. We also work on giant unilamellar vesicle, and this group is called GUV group. And in this group, we aim to functionalize GUVs and vesicles so that it can grow and divide, or it can form multicellular structure, or it can form cytoskeleton-like structures, and also will acquire molecular transport ability. And we also work on directed evolution and also basic protein science that is important to understand and also finally generate proteins that can be used to synthesize artificial cells in the lab. So if you find some interesting topics, please feel free to contact me, and I'll happy to talk to you and wish you that you can join our group and do research together. Thank you.
Hello, so I'm Naohiro Terasaka, an API in ELSI. So my group started in 2022, and currently so we have one postdoc, 3 PhD course students, 1 master course student, and 1 technical staff. Our research question is how functional biomolecules emerged and evolved in ancient Earth. To answer these questions, we are focusing on directed evolution of biomolecules. Directed evolution is a method mimicking natural Darwinian evolution. In general, firstly, we prepare parent gene and make library containing various sequences by diversifying parent gene. Then selecting improved functional molecules and convert the sequence information to DNA. By repeating this cycle, we can artificially evolve molecules in laboratory. The first research topic in my group is artificial cell and organelle. During last decades, it became possible that homogeneous picoliter emulsion is easily prepared and have been applied for fluorescent-based sorting machine by microfluidics technologies. We are trying to apply these techniques to make artificial cell and organelle to develop new type of directed evolution system. Second topic is primitive translation project. As you may know, RNA is considered as first functional biomolecule in ancient Earth. We are working on catalytic RNA ribozyme, especially on amino acylation reaction to develop primitive translation system and genetic code reprogramming techniques to utilize non-canonical amino acids. The third topic is synthetic virology. We recently evolved bacterial protein to virus-like protein capsule packaging RNA. We are trying to develop more functional virus-like structures to shed light on origin of virus research and virus alternative technology development. For more details, please visit our group website or directly contact me. Thank you for listening.
Hi, I'm Tetsuhiro Hatakeyama. Our group studies theoretical biophysics and systems biology and universal biology. And in particular, we'd like to know the theory for universal properties of biological systems. Universal property is mean property commonly conserved to the all living things, including bacteria, animal, fungi, plant, and human. And also the all living things, including not only the existing lives, and also primitive lives, kind of origins of lives, and also the extraterrestrial lives, something that the biosignature in exoplanets. So, by studying the universal property, we'd like to connect the existing life to the origin of life and life signatures. And we aim to understand life, not restrict to the materials currently used by the living organism, such as specific protein or specific DNAs. And our study is a theory, and you should run mathematics, physics, and computation. But if your major is biology or geoscience, by studying this topic, you can do this research. So please join us.
Hello, my name is Mohit Melwani Daswani, and I'm an associate professor here at the Earth Life Science Institute. by now you probably already know that ELSI is a pretty special place. So what I study is whether other worlds in our solar system could be or could have been habitable. And I do that by trying to understand how their interiors work. So the question sounds simple. Does liquid water exist somewhere beyond Earth, and could life survive over there? But to answer this question, you have to think like a geochemist, a geophysicist, and an astrobiologist all at the same time. And that intersection is exactly where my lab operates. So take Europa, for example, one of Jupiter's many moons. We know it has a global liquid water ocean beneath many kilometers of ice. But what we don't know is what that ocean is actually made of, how acidic or basic it is, and what sources of chemical energy it might offer to life. My approach is to model what happens to the rocky interior over geologic time, how it evolves. Minerals can dehydrate or hydrate, volatiles and chemical species can move around, and those fluids then can migrate upward to build the ocean from the inside out. We showed that some of these geological processes could have generated Europa's ocean, and we predicted an ocean rich in carbon dioxide. Later observations by others using the James Webb Space Telescope confirms that Europa's surface has carbon dioxide, which was maybe a validation of this type of framework. So we apply these same coupled thermal chemical models to worlds like Ceres and Titan, and we're now extending this framework to exoplanet contexts. In my lab, we combine thermodynamic computation, reactive transport modeling, and lab experiments where possible. We also connect directly to spacecraft data and sample return missions. So ELSI is a uniquely interdisciplinary environment. My group interacts with biologists daily, chemists, planetary scientists, and we're all working on these big questions that range from the origin of life to the design of future missions. So if you're drawn to these big questions about habitability and you want to develop both computational and experimental skills, I'd encourage you to get in touch. Thank you, and I look forward to hearing from you.
Hello, I'm Javier Martin Torres, a PI at ELSI. I'm going to describe you very briefly my past experience and research. You can find more information about myself in this link here. Well, imagine standing in a lab where we can test hardware that will actually fly to another planet. Those are exactly the kind of activities that we will develop in my team. My research stands at the center of planetary exploration and astrobiology. I study the habitability of the planets, and for that we send instrumentation to measure the variables that we need to understand these conditions. So we try to answer questions like, is Mars habitable now? I mean, can currently microorganisms populate the planet? Was there life in the past? And another even more difficult question, if there is life, can we find it? So I investigate the conditions for life on Mars, and for that I studied transient liquid water, brines, radiation, how organic molecules and even DNA fragments can survive in rocks. And yes, I am interested in life detection at ultra-low concentration, because if there is life now, or there was in the past, we expect very tiny amounts of material, either in situ sampling or from returned samples. I have been deeply involved in NASA's Curiosity rover as a co-investigator and science operations lead for the REMS instrument. REMS stands for Rover Environmental Monitoring Station, and it's the instrument that provides temperature, pressure, relative humidity data every day. So every time that you see those data, they are coming from those data from Mars, they are coming from our meteorological station REMS. And this is the instrument that delivered the first evidence of transient liquid water on Mars in a paper that we published in Nature Astronomy. So look at this spectacular graph from Curiosity rover. This is daily pressure, temperature, and humidity, and also UV radiation. And we have been obtaining this data from more than 15 years now. So it's amazing, and it's presenting us Mars much more dynamics than we have ever imagined. So developing instrumentation that go to another planet give us a lot of information that you can combine with your models in order to understand better the planets. I participate in several Mars missions that are operating on Mars and in orbiters. And I'm the principal investigator of the instrument HABIT, that stands for Habitability, Brines, Irradiation, and Temperature. And that is an instrument that is designed to measure UV, temperature, dust on the Martian surface, and also to produce liquid water on Mars. And something very, very important, our Habit flight model arrives to ELSI next month. And here are our latest breakthroughs. We have just published that fragmented DNA can survive more than 100 million years of Martian radiation levels and still be extracted and sequenced. We have grown microorganisms in real Martian regolith simulant at ultra-low water activity, and we have pioneering an instrument called Space Ion. This is a nanopore sequencer of DNA at the picogram level, and this instrument that acts without amplification will be very helpful for future JAXA and NASA missions. Space Ion is a DNA sequencer that more or less will look like this. And here are the news that I am most eager to share with you. We have just installed a brand new ISO 5 clean room at ELSI-2. This state-of-the-art facility will let us handle Martian regolith samples under planetary protection protocols, develop and calibrate space instrumentation like HABIT and Space Ion, and run cutting-edge astrobiology experiments in controlled, contamination-free conditions. It's a game changer for hands-on work that directly supports missions like the Mars Sample Return and future human exploration. We are perfectly placed for interdisciplinary breakthroughs, linking habitability models with real mission data. So if you're excited about physics, engineering, hardware that will fly to Mars and answer questions that are we alone, please contact me. This is my email address. Thank you.
Hi, my name is Soyoung Park. Welcome to Spark Lab. What is life? To answer this fundamental question, we investigate the origin and principle of life from the perspective of chemistry. Life is a system built from diverse biomolecules, and biomolecular interactions generate remarkable functionality and complexity. Among these biomolecules, we are particularly fascinated by nucleic acid. We design and synthesize biomolecular ensemble based on the programmability of nucleic acid. A hallmark of our research group is that we create new molecules with our own hand and use them as tools to investigate biomolecular systems. To achieve this, we pursue a highly interdisciplinary research group at the interface of chemistry, biology, and immunology. Let me introduce briefly our main research project. The first is the development of DNA-based hybrid catalyst. We have established the chemical strategy to incorporate amino acid into DNA scaffold to generate a DNA metal enzyme. Based on this approach, we develop a novel DNA nanozymes by recreating catalytically active site of complex protein within a compact DNA scaffold. We also focus on the design and application of fluorescence nucleic acid. We have developed a chemically modified nucleic acid and applied them to visualize the structural changes in nucleic acid and their interactions with the proteins. In particular, we are interested in left-handed helical structure, namely gDNA and gRNA. As you know, DNA and RNA usually adopt right-handed helical structure. However, under specific conditions, they can form a unique the left-handed zigzag conformation. Because a left-handed helical structure are highly transient and difficult to observe, we are developing fluorescence molecular tools to detect the interaction between left-handed nucleic acid and their binding proteins. And chemically modified nucleic acid represent a fascinating research area. Beyond their application as a functionalized molecule, they can provide a promising platform for expanding the chemical diversity of genetic information and exploring new biological systems. Spark Lab welcome students and researchers from a diverse research background. If you are interested in our research, please feel free to contact me. I hope we can work together and explore new scientific discoveries. Thank you.
Now, let's move on to the final topic, the application and entrance examination. On the left, you'll find the ELSI course webpage. Make sure to check out the website before you apply. On the right, you will find the official website for the International Graduate Program, IGP-C. The next call for the application will start in August. From the QR code, you can check the details for each contents. Here's a brief overview of the upcoming schedule. Orientation webinars will be held on July 24th, Japan time. From August to September, applicants should request a consent letter from one principal investigator. The deadline for submitting the consent letter is at the end of October. The application deadline is in mid-October. The entrance exam, which will be an interview, will likely take place in November. Finally, in December, applicants will receive the notification of results. Please note that exact dates will be announced in August through the IGP-C website. As for the letter of consent, you must submit the letter usually about 1 week prior to the application deadline. To obtain this, you should first identify an LCPI or API whose research interests align with yours. Second, then reach out to them by late September to discuss your research interests. Third, if both sides agree, request a consent letter from one PI. Please note that due to limited enrollment capacity, you may not receive a consent letter even after contacting a PI or API. After submitting all required documents, shortlisted applicants will be invited for an online interview, likely taking place in November. The full application procedure is detailed in the official application guide available online, as shown in the previous slide. Applications are submitted entirely online. Although the guide may mention a written examination, please note that for the ELSI course, there is no written examination, only an interview. The online interview will be conducted in English. Please prepare a 10-minute presentation with slides, followed by a 35-minute question and answer session. Questions will be focused primarily on your research presentation. In addition, some questions may be asked to assess your basic knowledge from your undergraduate studies. So that is all for the YouTube session. Thanks for watching, and we all look forward to your application.